Hi everybody, welcome to this lecture in molecular biology, where we will start learning about experimental tools for molecular cloning. We're starting with the first section of Chapter 4, which gives an introduction to gene cloning. What is cloning? Cloning can refer to the propagation of identical organisms. If there are any botanists in this class, you might be familiar with the concept of cloning plants from cuttings. These are parts of an original plant from which you can make clones. The word clone means twig or small branch in Greek. To clone a plant, we would take a cutting from the plant that you want to clone, and then what you do is you graft it onto a trimmed base or rootstock from another plant that serves as a vehicle to propagate a new genetically identical plant. So, the idea behind molecular cloning is analogous. In molecular cloning, you clone molecules of DNA, and what you do is you cut out a piece of DNA that you want to clone and you attach it to a vector, which is a vehicle to propagate copies of the cloned DNA. Here, in this diagram, this is a plasmid vector. You take your cut DNA fragment and you attach it to your vector. Then you introduce the vector with the DNA fragment into a cell, typically a bacterial cell, and then you allow the DNA in the vector to replicate within the cell. This happens by the DNA replication mechanisms that we discussed in Chapter 2. Then you can allow the cells to multiply, and then you can isolate the clones. You can culture bacteria from colonies that derive from individual cells that had been transformed by this DNA, and you can isolate many copies of it. We have shown in this slide a plasmid that can be used as a vector. A vector is a carrier of DNA. These are often plasmids. Plasmids are circular DNA molecules. They are small in comparison to bacterial genomic DNA, and they replicate independently of chromosomal or genomic DNA. You could have many copies of plasmids replicating in a bacterial cell. Plasmids are a very common vector for cloning DNA, and shown here is a very commonly used plasmid called PUC18. Common characteristics of these plasmids are they are circular DNA molecules, they have an origin of replication so that they can replicate by themselves independently of chromosomes. When these plasmids are used as vectors for cloning DNA, they will have a selectable marker, which is often a gene that confers resistance to an antibiotic, so that you can select for bacterial cells that have taken up the plasmid along with the cloned DNA by culturing them in the presence of that antibiotic. Only the cells that have been transformed with the plasmid will be able to grow. Other selection markers could confer the ability for the bacterium to use a certain nutrient that it otherwise wouldn't be able to use. So if you are growing it on a certain medium that is absent of other sources of nutrients but includes this key nutrient, then that could be a selectable marker. Often these DNA vector plasmids have what are known as multiple cloning sites. These are regions of DNA that are recognition sites for restriction endonucleases. What are restriction endonucleases? Restriction endonucleases, also called restriction enzymes, are bacterial DNA cutting enzymes that were discovered in the 1970s. These enzymes specifically cut DNA at precise sequences. Their discovery won these scientists, Arbour, Smith, and Nathans, the Nobel Prize in 1978. Restriction enzymes exist in bacteria as a defense mechanism to chop up foreign DNA, and they are part of the bacterial immunity that protects bacteria from viral invaders that try to inject their own DNA. These enzymes are called endonucleases because they cut in the middle of a DNA sequence, in contrast to exonucleases that digest the ends of DNA molecules. Restriction endonucleases recognize specific sequences. Here we see some examples of recognition sequences for different restriction enzymes. What can we notice about these sequences? Is there any particular pattern? You may realize that these are palindromes. What is a palindrome? A palindrome should be read the same way forward as it is backwards. For example, GGATCC. How is this a palindrome? We are reading this 5' to 3' on one strand, and if we read 5' to 3' on the complementary strand, we will also see GGATCC. But this table only shows one strand of the double-stranded DNA that each of these enzymes cut. So this is the recognition sequence for the BAMH1 enzyme. And this is the recognition sequence for the EcoR1 enzyme. So if you read 5' to 3' on this strand, it is the same sequence as if you read 5' to 3' on the antiparallel complementary strand. Restriction enzymes thus recognize specific sequences that are typically palindromic, as we just described. They cut both strands, breaking the phosphodiester bond between the nucleotides indicated here. And now you have a double-stranded break, and then these two DNA molecules can come apart, and they have overhanging regions which can act as what are called sticky ends, which I'm going to talk about again later. But first, I'm going to talk about methyltransferases. In addition to having restriction enzymes, bacteria have evolved corresponding enzymes that protect their own DNA from attack by the restriction enzymes that they produce. These are methyltransferases. They modify DNA by adding methyl groups to specific bases. So this prevents the recognition of those sequences by restriction enzymes that otherwise would cut those sequences. Usually, restriction enzymes do not recognize DNA sequences with a methyl group on either strand. When the bacteria replicate their DNA, it remains protected from restriction enzymes because the semi-conserved daughter DNA has one strand that was previously methylated. In this example, both parent and daughter double-stranded DNA is protected from cleavage catalyzed by EcoR1, which can only cut at its recognition sequence if both strands are non-methylated. After DNA replication, methyltransferase adds a methyl group to new daughter strands, which results in a methyl group on both strands. So when the DNA replicates again, one strand in each replicated DNA will always be methylated. And this system keeps the bacterium's own DNA protected from its own restriction enzymes at all times. There are other cases where the situation is reversed, in that the restriction enzyme recognizes the methylated DNA and not unmethylated DNA. But what is shown here in this slide is a common example of how bacteria protect their own DNA from endonucleases. Restriction enzymes and methyltransferases make up the two-part system known as the restriction modification system. The restriction enzymes from this bacterial defense mechanism can be useful for editing DNA in the lab. These restriction endonucleases, along with an enzyme called DNA ligase, provide tools for molecular biologists to cut and paste DNA. The restriction enzymes are analogous to molecular scissors, with which we can cut DNA at specific sites, and DNA ligase can glue cut pieces of DNA together. As I said, many restriction enzymes cut double-stranded DNA in such a way that they leave sticky ends. Two DNA molecules that are cut with restriction enzymes that leave the same overhanging, single-stranded region have compatible sticky ends that you can bring together by base pairing. These are very short regions of single-stranded DNA, so this base pairing interaction is very transient. That is, the forces holding these strands together are weak, and the hybridization is short-lived. But with the sticky ends loosely bound, you can join the backbones of these strands together using DNA ligase. It is also possible to stitch DNA pieces together that do not have these sticky ends, but instead have blunt ends, but the ligation of sticky ends is much more efficient. Shown in this slide is the reaction catalyzed by a restriction enzyme in this direction, cleaving a phosphodiester bond by hydrolysis, and in the other direction is shown the reaction catalyzed by DNA ligase, which uses the energy from ATP to form a phosphodiester bond. Restriction endonucleases and DNA ligase are very useful enzymes in the lab. With these tools, it is possible to cut and paste DNA. The first demonstration of this, shown here in this illustration, was performed by Boyer and Cohen in the 1970s. They took two naturally occurring plasmids in E. coli. Each of these plasmids had resistance to different antibiotics. One of them, PSC101, is resistant to tetracycline, and the other, PRSF1010, is resistant to streptomycin. Each of these plasmids had one recognition site for the EcoR1 restriction enzyme. They cut these plasmids with EcoR1 and stitched them together with DNA ligase, and then used the recombinant plasmid to transform bacteria. What is shown here in this diagram is these two plasmids coming together as one. But there are other possible scenarios for this. These two plasmids have EcoR1 sites. The idea is you cut them so they are linear fragments of DNA with compatible ends, and then we ligate them together into a recombinant plasmid. But it is also possible, and probably more likely, that the two ends of the same fragment come back together and ligate, resulting again in the original PSC101 and PRSF1010 plasmids. How do they tell whether the bacterium was transformed with this plasmid or either of these plasmids? How do they tell which piece of DNA went into the bacteria? In the recombinant plasmid, there are two resistance markers here, coming from each of the original parent plasmids. So to select for the plasmid that has both, the bacteria are grown on plates that contain both tetracycline and streptomycin. Couldn't bacteria be transformed with both of the parent plasmids simultaneously and become resistant to both antibiotics without taking up the recombinant plasmid? Well, this transformation process is a relatively rare event, meaning that most cells will not take up any of these pieces of DNA, and maybe 1 in a million cells will be transformed by taking up a DNA molecule. It's much more rare that an E. coli cell takes up more than one DNA molecule. So, in theory, this can happen, but it typically doesn't. So, we saw that with these vectors that are cut in order to put DNA into them, we can have the problem of these coming back together without your piece of DNA being inserted, and that the empty vector could be introduced into your E. coli cells. The solution in this case was that you can have two selectable markers, one of them on the piece of DNA that you want to introduce into your vector. What if you can't do that though? What if you want to clone a piece of DNA that has no selectable marker? You do not have a way of selecting for that DNA. So one solution illustrated here is a method to prevent your vector DNA from religating to itself. DNA ligase catalyzes the formation of a phosphodiester bond between the 3' OH of one single strand of DNA to the 5' phosphate of another. So you can remove this 5' phosphate using an enzyme called phosphatase to prevent self-ligation, and that phosphatase-treated vector can be ligated with an insert that has its intact 5' phosphate groups. This is what DNA ligase does. It forms the phosphodiester bond between a 3' OH group and a 5' phosphate group. To show this more schematically, we have two sticky ends coming together. The DNA ligase is forming the phosphodiester bond using the 5' phosphate. So if you remove the phosphate groups here, DNA ligase will not be able to form the phosphodiester bonds with the two ends that have had their 5' phosphates removed. But now, if you introduce an insert that has a 5' phosphate on its 5' end, then the DNA ligase will form a phosphodiester bond on that strand and leave a nick on the other. This happens on both ends of the DNA that is being inserted into the vector. That will keep the plasmid together, and it can be used to transform E. coli, and the bacteria will repair the nick intracellularly. In this way, you can prevent your cells from being transformed by empty vectors with no inserts, and favor the transformation with a vector that has a DNA insert cloned into it. Another method for doing this is called directional cloning. You can use two different restriction enzymes that produce non-compatible ends. A vector that is digested so that it has two incompatible ends is unlikely to self-ligate. You can use this twice-cut plasmid vector and insert your cloned DNA that has also been cut with two restriction enzymes, producing sticky ends that match the two distinct sticky ends of the twice-cut plasmid. So in this way, you can both prevent the self-ligation of the vector, and you can also ensure that you introduce the insert in a specific orientation. Once you have attached the DNA insert into a cloning vector, how do you get it into E. coli? How do you introduce it into the cells? There are a few different ways to do this. These are two common methods to introduce recombinant DNA into cells for cloning. by heat shock of chemically competent cells, or by electroporation. For heat shock, cells can be made receptive to taking up DNA by chemical treatment. We call these cells competent since they are able to take up DNA. Cells can be made competent by incubating them in a buffer solution containing calcium chloride. Calcium ions bind and destabilize the cell membrane, and also the positive charge attracts negatively charged DNA molecules, and by doing a short incubation, heating at 42 degrees Celsius or so, that causes a disruption in the cell membrane and allows DNA to go in. This is how heat shock transformation of chemically competent cells works. In electroporation, you apply an electric field to a suspension of cells in a non-ionic solution. You use a quick pulse of electric potential, and it opens up pores in the cells, and then that allows your DNA to come in. When you carry out this procedure, either by heat shock or electroporation, you have your cells in a tube. You transform them and plate them onto agar with selective antibiotic, or whatever you have a selection marker for. You let the cells grow, and then you isolate colonies. So as I said, the event of DNA getting taken up into the cells is rare. You have maybe 1 million cells and 1 billion DNA molecules in the mix. Maybe only 100 cells or so will be transformed. Most cells do not take up any DNA, and the cells that do take up DNA usually only take up single pieces of DNA. So you plate these cells and you physically separate them so only the single cells that have taken up the DNA will grow and multiply to form a colony. Each of the cells in a colony has multiple copies of the same piece of DNA that was taken up by the original transformed cell. Typically, only circular DNA containing an origin of replication will transform bacterial cells. Linear pieces of DNA will not replicate properly, and they will be digested by exonucleases inside the cell. How can you identify which of the colonies on your agar plate have taken up the target DNA inserted into the vector? We were talking about a couple of methods to improve the likelihood of colonies growing that have the insert, but you want to make sure that you are screening clones that have the pieces of DNA that you want. Shown here on this slide is an example of one method for identifying clones that have an insert. In this example, the plasmid PBR322 serves as a vector. You have two selection markers here on the vector, ampicillin and tetracycline. You insert your piece of DNA into the vector by cutting the vector at a restriction site, which is inside the ampicillin marker, and then you ligate the DNA fragment into the cut vector. Then you transform your cells, and you can look for which clones have been disrupted in that selection marker. In this case, you have the vector which has been cut by the enzyme PST1. The insert was also cut by PST1, and then the two are ligated together. Some of these plasmids are going to self-ligate. Then you transform bacteria, and you grow on tetracycline, and then you make a replica plate onto plates with ampicillin. Some of the colonies that grow on tetracycline will not grow on ampicillin. So which are which? Which cells have the clones containing the insert? They're the ones that did not grow on ampicillin, but did grow on tetracycline. What are the other ones that grow on ampicillin and tetracycline? They are the clones with the self-ligated vector. This is one method for identifying clones. A more common method is called blue-white screening. This is a way to visually identify which clones have the desired insert. PUC18 cloning vectors carry a gene coding a polypeptide subunit of the galactosidase enzyme LAC-Z. Galactosidase activity can be complemented in cells that otherwise lack this subunit when they are transformed with PUC18. Within the region coding this polypeptide is a multiple cloning site that has several restriction sites into which you can insert a piece of DNA. If you insert DNA into this site, that disrupts the coding sequence of the galactosidase subunit. Then you can detect the disruption by observing the lack of galactosidase activity on a chromogenic substrate. This substrate turns blue when it's turned over by the galactosidase. This is XGAL. If it is hydrolyzed by beta-galactosidase, it releases this leaving group, which then dimerizes to form the blue indigo dye. You can tell which clones have galactosidase activity by observing which clone is blue. In this case, we are trying to insert the piece of DNA into this vector within the coding region for the galactosidase subunit. So if we transform an E. coli strain that requires the gene on PUC18 coding the LACZ subunit for galactosidase activity, which of these colonies contains a vector with an inserted piece of DNA? Are they the blue colonies or the white colonies? Cells that take up a plasmid with an intact region coding this LACZ subunit will have galactosidase activity, and so they will turn blue, because they hydrolyze XGAL to produce the blue indigo dye. What happens if you insert a piece of DNA into the multiple cloning site within the gene coding the LACZ subunit? What happens to the galactosidase activity? You get no activity, and so no blue color with XGAL. In this blue-white screening, we are looking for white colonies, which have a plasmid vector with an insert disrupting the LACZ gene. I will pause here. So far we have discussed plasmids as vectors for molecular cloning. In the next part, we will talk about vectors derived from bacteriophage.